Okay, awesome. So yeah, as Simon said, I'm going to tell you about my PhD work in the Shapiro lab at Caltech, which was focused on trying to enable ultrasound imaging of engineered probiotic bacteria in vivo. So first of all, kind of the motivation behind this. Why do we want to be able to image to be able to image bacteria inside the body with ultrasound? It's because there's a lot of interest in using engineered bacteria as new therapeutics and diagnostics, such as using tumor homing bacteria to treat certain kinds of cancers, and using uh, engineered gut bacteria to diagnose certain gastrointestinal conditions like gut inflammation. But kind of where we saw the gap is that once the bacteria are inside the body, we didn't really have many good tools to be able to monitor their location or what they were doing. So in terms of methods that people commonly use to image bacteria inside the body, so most methods are based on optical methods like using bioluminescent or fluorescent reporter genes like GFP. And this is because they're really easy to use. You just need to put one gene in your bacteria of interest. But unfortunately, where animals and humans are not transparent, we're pretty opaque, so light can't really get through tissue well. So you're very limited in terms of imaging depth and especially resolution at depth. So to get around this, people have made nuclear imaging methods such as PET and SPECT that rely on specific radioactive tracers that are specifically uptaken by the bacteria. And this provides pretty great sensitivity and imaging depth, but unfortunately you have to use radioactive tracers, which are difficult to implement. So we'd much rather use something like ultrasound imaging, which is a relatively cheap and fast imaging modality that gives you good imaging depth. I'm sure you've all seen images like this before. But up until recently, we didn't have a way of seeing bacteria under ultrasound imaging. That was until these things called gas vesicles came along. So what gas vesicles are these hollow protein nanostructures that have a protein shell, and their interior is entirely filled with air. And they're found in nature in these aquatic microbes, such as cyanobacteria and haloarchaea, which make these gas vesicles to regulate their buoyancy for optimal access to light and nutrients in the water layer. And how they make them is that they have these large clusters of many genes, where the first gene, GVPA, is the primary structural component that encodes for a very hydrophobic protein that is so hydrophobic, in fact, that as it's forming the shell, it is able to exclude water from the interior so that it's entirely filled with air. So our idea is that we take these gas vesicle genes and put them in our probiotic bacteria of interest so that we can see them under ultrasound imaging. And so then the question is that how do these enable us to see bacteria under ultrasound imaging? So to understand that, I'll just go through sort of the basics of ultrasound imaging. So how it works is that you start with an ultrasound transducer, which has these piezoelectric elements, where if you apply electrical pulses, they convert this into sound waves that is very high frequency, on the order of megahertz. And these sound waves go into your tissue and interact with it. And they can interact with it by transmitting through and also reflecting back. And the transducer can actually pick up on these reflected waves back. And based on the time it took the waves to transit, and also how fast they were going, which is a known quantity, the speed of sound in your tissue, you can calculate the distance of this object in the tissue, and do that a whole bunch of times with a whole bunch of fancy signal processing methods to actually reconstruct your image. And the reason gas vesicles work so well here is that they have this air-water interface that really strongly reflects the sound waves back. So we can actually see the bacteria under ultrasound imaging if they're expressing gas vesicles. But kind of the issue with this is that both tissue and gas vesicles have the same sort of signal. So then the question was, how do we separate out these two? So we want to be able to image gas vesicles specifically under ultrasound imaging to separate them from the tissue signal. And so how this works is it's based on one of their properties, which is that they can be collapsed under high acoustic pressures. So you can start off with your intact gas vesicles, which look like this under electron microscopy. And then at a certain time, you can apply a high acoustic pressure, which causes them to collapse. And while they're collapsing, they release these little tiny air bubbles, which cause a transient, very high spike in the ultrasound signal, which you can process out and get the GP specific ultrasound contrast, which is separate from the tissue, which has this sort of linear response and just stays high in response to the high pressure. And so throughout my talk, I'm going to be talking about the GV-specific ultrasound signal, and this is what I'm referring to based on collapse, which is very sensitive and allows us to separate the GV signal from the tissue signal. And so when I first joined the Superior Lab, there were initial gas vesicle gene constructs that worked to some extent in probiotic bacteria, but they didn't work too well. They were based on gas vesicle genes from Anabina and Bacillus. And so they just didn't express very well, especially not at body temperature. And they also were very burdensome to the cell and toxic. They caused a lot of growth defects and kept escaping. And so all these factors led to them not being able to be expressed in situ. So in order to get these sort of initial in vivo results from the lab, the bacteria had to first express the gas vesicles under ideal conditions in culture, and then they were injected into the mouse, in this case in the colon, to be imaged. But this isn't very relevant for monitoring bacteria inside the body. We want them to actually be expressing the gas vesicles in situ and then be able to see them. So kind of the first step in all of this was optimizing gas vesicle expression in a probiotic bacterium. And for this, we chose the widely used probiotic bacterium, E. coli Nissl. And the approach we took here was a sort of a genomic mining approach where we took gas vesicle genes from a variety of different microbes known to express gas vesicles and tried them in E. coli. And you can read more about this in the paper, but long story short, the gas vesicle genes from this strain of Serratia worked the best for expression in E. coli. And then for porting it into E. coli Nissl, we had to do some optimization of the backbone. So we put it on a low copy origin backbone with arabinose induction and a stability cassette. And in this case, we were able to get very high levels of gas vesicle expression, where you can see these E. coli Nissl cells were very buoyant when we centrifuged them. And when we looked at them under phase contrast microscopy, we could see that they were, most of them were pretty full with gas vesicles, which you can also see under electron microscopy. So once we had that working, we wanted to see, okay, can we see these cells well under ultrasound imaging? So that's what we did next, and what's plotted here. So it's the gas vesicle specific ultrasound signal versus varying concentrations of these gas vesicle expressing Nissl cells, and also fluorescent protein expressing cells as a negative control. And at the bottom, you can see the cross sections of the agarose phantoms that we're actually imaging. And fortunately, with this gas vesicle specific imaging method, we can detect down to 10 to the 5th cells per mil under these ideal in vitro conditions. So once we had that working, we wanted to go in vivo in the first main application, which is in tumor homing bacteria. So we got the engineered Nissl to home to tumors in mice by injecting them in the tail vein, and then we injected the mice with arabinose to induce the gas vesicle expression, and then did some ultrasound imaging to look for the bacteria inside the tumors. So these images in gray at the top here are the B-mode images, which show the anatomy of the tumor. And these images in blue here are the gas vesicle specific ultrasound signal. And in all the mice that had tumors colonized by the gas vesicle expression Nissl, we saw strong GV specific ultrasound contrast in the necrotic core, where the bacteria are known to colonize because the immune system cannot clear them from this area. And you can maybe see this better in the overlay. And also another really good thing about ultrasound imaging is that you can not only get these 2D cross sections, but you can actually get 3D images if you scan the transducer over the entire tumor and then stitch these 2D images together, which is what is shown here. And so we were really excited to see the gas vesicle specific ultrasound signal distributed throughout the interior of the tumor. And in this case, they were actually directly injected into the tumor, so you can actually see the injection site, just confirming that we are able to see the engineered bacteria inside the tumor in real time, which is important for being able to monitor these tumor homing bacterial therapies. So that was in tumors. The other main application is inside the gastrointestinal tract. If these slides will go forward, here we go. So yeah, the other main application is inside the gastrointestinal tract. So next we got the engineered Nissl to colonize the gastrointestinal tract of mice using antibiotics. And we were able to see strong GV-specific ultrasound contrast in all of the mice that were colonized by the GV-expressing Nissl and not in the control mice that were colonized by a fluorescent protein expressing Nissl. And these are actually 2D projections of 3D acquisitions where we scan the transducer across the entire abdominal area of the mice. And so we can also display this data in 3D, which is what is shown here. And we also wanted to confirm that where we were seeing the bacteria. So it looked like from these in vivo images that the bacteria were mostly in the cecum and the colon, which is the large intestine of the mice and where the bacteria are known to colonize. So to confirm this, we did some ex vivo imaging where we took out the intestines and then scanned the transducer over the intestines. And you can see strong GB-specific ultrasound contrast primarily in the cecum and the colon, which is where we expect the bacteria to colonize, just confirming that that is the location that we're seeing in the in vivo images. And finally, we're not just limited to in vivo and ex vivo imaging. We can also detect the GB expressing Nissl in the feces, just kind of showing the versatility of this method. And so once we had GB expression working in the gastrointestinal tract, we wanted to do some sort of diagnostic application. So next, we aim to use these engineered bacteria as sort of probiotic acoustic biosensors to be able to do ultrasound imaging of intestinal inflammation. And so how we envision this is that the patient would consume a diagnostic yogurt containing the engineered bacteria, and they would temporarily colonize the gastrointestinal tract. And when they are sensing some sort of inflammation marker, they would express the gas vesicles that we can see with ultrasound imaging. So how the bacteria would do this is that we would engineer them with these 2-component systems that consist of a membrane sensor kinase protein that when it binds to its ligand, it's able to phosphorylate the second component, the response regulator, which then activates transcription of a specific promoter. And we can put this promoter upstream of the acoustic reporter gene, so the gas vesicle gene, so that we get gas vesicle expression in response to sensing the inflammatory biomarker that we can see under ultrasound imaging. And so what these biomarkers are these oxidized sulfur and nitrogen species, which our collaborators showed are kind of elevated during intestinal inflammation because there's a lot of reactive oxygen species during intestinal inflammation, either from the host cells or from bacteria themselves. So what I'll focus on now, since I have limited time, is our thiosulfate sensor. And this is just because it worked well. So the initial thiosulfate sensor is shown in this plasmid map where we're constitutively expressing the 2 components, so the membrane sensor kinase thiosulfate S, which binds the thiosulfate, and also the response regulator thiosulfate R, which in the presence of thiosulfate activates this promoter, and we put it upstream of the gas vesicle genes from Serratia so that we would get gas vesicle expression in response to the inflammatory biomarker thiosulfate. And so this initial construct turns out didn't work too well, so I had to do some rounds of mutagenesis and screening to improve the sensor. So the kind of workflow for this was to create mutant libraries, screen them in E. coli, and then a nice feature about gas vesicles is that if you get high gas vesicle expression in your cells, the colonies on plates will look very white by eye. So I was able to just basically pick the colonies that were very white on plates with thiosulfate and then go forward with them and characterize them in patch format and calculate the patch opacity, which is a measure of gas vesicle expression, to be able to compare the top variants. So this is a summary of the mutagenesis and screening that I did to try and improve the thiosulfate sensor, especially because it wasn't working well at body temperature. You can see the parent here, it worked fine at 30 degrees Celsius, but it didn't work at all at 37 degrees Celsius. And what's plotted here is the relative patch opacity, which is a measure of the gas vesicle expression. So the first thing that I did was doing site-directed mutagenesis on the 2 constitutive promoters that were expressing the 2-component system components. And I was able to get some marginal improvements, but not by much. And then we expected that this membrane sensor kinase protein was just not being expressed well. So next I did error-prone PCR on the sensor kinase protein itself. And in this case, I was able to get one mutant that performed very well at both 30 and 37 degrees Celsius, which you can probably see better here. It had a very high patch opacity at both temperatures, and so it was much better than the parent. And it turned out that it had these 4 point mutations throughout the protein. And so we tried this optimized thiosulfate sensing variant in vivo, and unfortunately it still didn't work. So we tried a bunch of things to improve the sensor even more, and it turned out that the best thing for it was to actually implement this recombinase-based switch, where I put this integrase downstream of the thiosulfate activated promoter, and then put a strong constitutive promoter upstream of the gas vesicle genes, so that in the presence of thiosulfate, the cells will express the integrase and flip the direction of this strong constitutive promoter, so that I'll get strong constitutive GB expression in response to thiosulfate sensing. And so it turns out that when I add this integrase switch, it really boosts the signal by more than 2-fold and also increases the sensitivity. I also made a GFP version of this recombinase-based switch and confirmed that it does actually improve the sensitivity and the signal, and also makes it have this switch-like behavior. So finally, this optimized thiosulfate sensor with the recombinase switch was good enough to test in a disease model. And so the disease model that we chose was streptomycin-treated mice, because it turns out that when you put high amounts of streptomycin in their water, they exhibit weight loss and also signs of intestinal inflammation that we can see under histology, such as edema and these inflammatory infiltrates. And I was also able to develop an ICMS method to actually measure the thiosulfate in the feces, and it turns out that streptomycin-treated mice consistently have it elevated above healthy control mice. And so the next step was testing our optimized thiosulfate sensing strain in healthy mice versus streptomycin-treated mice. And it turns out that only in the streptomycin-treated mice did we see elevated GB-specific ultrasound signal, which is what you can see in the quantification here. And we weren't able to see it in the healthy controls or in the GFP output control mice, confirming that we're able to do ultrasound imaging of streptomycin-induced inflammation in mice. So in conclusion, hopefully I've showed you that we were able to optimize gas vesicle expression in the widely used probiotic bacterium E. coli Nissl using these gas vesicle genes from Serratia. And we're able to image this engineered Nissl strain after it homed to tumors of mice and also colonize the gastrointestinal tract. And we're able to use these gas vesicles in a diagnostic application by linking them to thiosulfate sensing, so that we were able to image intestinal inflammation in antibiotic-treated mice. And then finally, I just want to thank everyone involved in this, especially my former PI, Mikhail Shapiro, everyone in the GB subgroup, my 2 really talented undergrads, Anne and Jamie, and of course, all the funding sources. And yeah, thank you all for listening, and I'm really happy to take any questions.